Good morning, everyone, and welcome to a new EBD seminar. My name is María José Ruiz, and I'm going to be introducing our speaker today. And afterwards, Irene Mendoza is going to be moderating the questions. Before I introduce the speaker, just a couple of reminders. If you need a certificate of attendance, please write down your name in the YouTube chat and then send an email to seminars@ebd-CSIC.es. Also, if you want to make any questions, you can write it down in the YouTube chat, and afterwards we'll be making them to the speaker. So our speaker today is Enrico Bazzicalupo. He is a PhD student here at EBD in the Department of Integrative Ecology. He's a second year PhD student and he has an FPI grant fellowship. Enrico, when you are ready, you can start. Welcome.
Hi, thank you, María José. So today I'm going to be talking about my PhD project, which is titled Speciation, Gene Flow, and Selection Genomics in the Lynx genus. So just to start off, I want to give a brief introduction on why I think genomics is a great tool to study different things. So first of all, genomics gives us the possibility to look at both past events as well as present ones. And this duality between past and present is also reflected in the duality between theory and practice. So genomics gives us the tools to both make theoretical advancement questions regarding evolution, speciation, and gene flow, as well as making more informed decision at the moment of doing practical decisions regarding population assessment or genetic rescues and more general conservation questions. So with genomics, if you have a model group, there are different things you can start looking into. One of the first things you can look at is demography. So how does the population size change through time in this group? Another process you can look at is divergence. So it's the process which brings the formation of different lineages from one group into two groups here. And you can also look at how these changes have, in effective population size, have been going on in these different groups. Another thing you can look at is a process called admixture, which is a way of saying that if any migration happens between these two populations, then the genes from this group will go into this other group if the migrating individual can reproduce. And this way, there is a mixture of the genomes of these two different groups through the process of migration. And finally, another interesting thing you can look at when using genomics is selection, which is the process through which an adaptive and an advantageous trait will become more and more frequent across a population through time. So in general, we can say that events of demography, differentiation, admixture, and selection can shape the evolutionary history of a group of species. And these processes leave like distinctive traces in the genome, which you can look for and harvest to learn about this process in the end. So our model study for studying all of these processes is the genus Lynx. So we have 4 different species in this genus. We have 2 American ones in North America, the Canada lynx in the north in Canada, and the bobcat in the southern part of North America and central part of North America. And then in Eurasia, we have 2 other different species. One is the Eurasian lynx, which is this widely distributed across all of Eurasia. And we have the Iberian lynx, which I don't know if you can see, there's very little distribution, but it's here in southern Spain. So what makes this genus a good model for studying genomics? Well, different characteristics for the different questions you have. So first of all, you can see with these big distributions in most of the species, but also small ones, you can say that the lynxes have very contrasting demography. And so we have low population sizes in one of the species, but also in some of the populations in one of the species, such as the western limit of the Eurasian lynx distribution, which has very fragmented populations. So this is with respect of demography. Regarding admixture, we can say that the fact that there are overlapping ranges between species gives the opportunity of admixture to take place if any of these coexisting individuals reproduce between each other. And so we have overlapping ranges both in the present in North America between bobcats and Canada lynxes, but also in the past we had overlapping distribution in the northern part of the Iberian Peninsula between the Eurasian lynx and the Iberian lynx. With regards to selection, this is a kind of interesting model because we both have very specialist species such as the Canada lynx and the Iberian lynx, which prey only on specific prey, while the other two species are very generalist in this type of ecological setup. And so we can look for these differences also, and which might give different selective patterns across species. But also, if we look at these wide distributions, we see that both within a species and between species, there are very different climates that go through them, which might create different selective pressures. So we have a good model. We have so we tried to sample it as best as we could. So we gathered a total of around 170 samples across the distributional range of these 4 species. And we extracted their DNA, and both in our lab and in collaboration with other people from other research groups. And after the DNA extraction, we sent the DNA samples to sequencing centers, which use the Illumina platform to generate whole genome sequencing data. So the sequence of the whole genome was gathered for all of these samples. And from those whole genome sequences, we can move on to the genomic analysis, which is what I'm going to be doing. So if we look at the objectives of my thesis, we can take the little graph I've showed you before and say that in the first chapter, we're going to be focusing on trying to model the demography, the divergence events, and the migration events in the genus Lynx to try to understand their evolutionary history. In the second chapter, we will focus on specifically on admixture to try to look and reconstruct the admixture events within the different species of lynxes and what consequences they have on the genome. And in the third chapter, we will be looking at the effect of selection. And so we will look at just one of our species, the Eurasian lynx, to try to discover the different drivers of selection and if this selection has any effect on population structure. So let's move on to the first chapter. So where we're going to try to build models of demography, divergence, and migration. Okay, so a bit of introduction on what we know. So if different research groups have tried to resolve the phylogenetic relationship between the different lynx species. And so in 2 studies, one from 1981 from Wenderling and another one from 1999 from Belinda Emmons, what they discovered is a sister relationship between the Canada lynx and the Eurasian lynx. So they use both morphological data and few molecular markers to reconstruct this phylogeny. But a more recent study, which uses additional markers both in the nuclear genome and in the mitogenome, try to reconstruct the same story, but they found a big difference. So in this additional reconstruction from Johnson et al. In 2006, we can see that the Iberian lynx and the Eurasian lynx are sister species, with the Canada lynx separating the 4. So this is kind of the situation up to 2006. But then in 2019, an additional study came out, which was, again, trying to reconstruct phylogenetic relationship between felids and lynxes also. So what they did was they used whole genome sequencing data this time. And what they found was like a kind of a dual signal between these 2 different models. But as they found this dual signal, they tried to focus on areas of the genome with low recombination rate, which translates into areas of the genome which are less affected by events of admixture. And what they found for those areas was this kind of relationship with the Canada lynx and the Eurasian lynx as sister species, which justifies the founding of the other model because of extensive admixture events between the Eurasian lynx and the Iberian lynx, which might have caused their genome to look so much alike that they might be found as sister species in other studies. So what we can take home from this is that admixture has played a central role in the shaping of the evolutionary history of the genus Lynx. And so what we try to do is try to build our demographic and divergence models where we try to reconstruct the divergent times between the different lineages and their demography, to be also aware of these admixture events which play a central role in the evolutionary history. We try to do this with a method called Approximate Bayesian computation with deep learning, and which was developed from a group called from Mondal et al. In 2019. And we are lucky enough to be collaborating with one of the persons which was on the development team. And so they took this model and they tried to make a few modifications to adapt it to our kind of data, and we could be looking into that. So just before I explain more or less how the model works, I just wanted to tell you what kind of data do you have to feed to the model. So our input data set was 8 samples from the 4 species, so 2 samples of each species. We use the cat as the reference genome for the alignment, and we used 4 samples to train the model, and the other 4 samples were used to validate it. And this was done with this initial 8 samples, but then can be repeated with the additional samples we have available to try to repeat the validation with some kind of replication. So the total data set was around 6 million SNPs from a total genome area of around 1 gigabase, which is 1 billion base pairs. So apart from your genomic data, you also have to input some parameters into this model, which are the average mutation and recombination rate across the genome, the generation time of your species, and also because you're trying to infer some output parameters, you have to kind of give a prior distribution or a range of values that you want to that you're trying to infer. And also because you are testing different models, you have to give the different topologies you want to test. So just very briefly, how does this method work? You can simulate data for genomic windows, and this will go through a deep learning algorithm, which will give you a kind of distribution of a summary statistic across all these windows you simulated or your actual data. Then you can take this distribution and pass it through another deep learning algorithm, and will give you a single summary statistic for your model. With this single summary statistic, you can do 2 things. First thing is to evaluate the model, so you can see how well the model is replicable and how your data compares to different models. So you can assign a model to your actual data. And also you can infer different parameters associated with the model, which is something we are very interested in, because the model will give us effective population sizes across time. They will give us the divergence times of the different lineages, as well as the amount of migration that happened between different lineages. So with that in mind, we tried to test the 2 different phylogenies that we discussed before. So the one with the Iberian lynx and the Eurasian lynx as sister species, and the one with the Eurasian lynx and the Canada lynx as sister species. And based on some preliminary results, as well as the idea that there might be some kind of extinct lineage inhabiting Europe that might have exchanged genes with our lineages, we also introduced ghost population, which is basically an additional lineage which is not sampled today, but still leaves a trace in the genomes of the samples you have, because of it has exchanged genes with them across time. So we're going to compare these 4 models, and the idea is try to understand which of these models has the highest support, and also once we know that, to try to know what the parameter values are. So when did the lineages diverge, how much did they mix between each other, and what were the changes in population size across time. So with that, we can move on to chapter 2, in which we were going to dive into the genomics of admixture. Okay, so once we have reconstructed our model for our genus, then we can take the individual genomes of each of our samples and try to, with different approximations, which might be window-based introgression scans, local ancestry, inference, coalescent models. What we try to do, and I try to explain this with very high-tech slides, is try to, what the program does is basically look at your genome and try to assign an ancestry to each area. So in this case, these red areas of the genome will be of the ancestry of this animal, while the blue ones might come from a different lineage. Once you have assigned these ancestries to all the areas of the genome, you can take this introgress, they are called windows, and try to ask some questions. So from the ancestry composition, you can basically ask yourself how much of the genome is coming from another lineage in one species, at what time did they come, and so are some genes introgressed more recently, are some more ancient, what kind of genes were introgressed in this process. Also, if selection is acting on any of those genes, and also if there are individuals from different geographic regions which have been affected differently from admixture. So maybe the westernmost part of the distribution shows more introgression than the easternmost part. And also if these patterns of difference can be also traced at the genome level. So are maybe highest areas of recombination less affected by admixture, or one chromosome more than another, this kind of questions. So unfortunately, they are just questions for now, but these are the types of things we're trying to look at in chapter 2. I don't have any results ready, so we can now move on to chapter 3, in which we are going to look at the effect of selection on functional and adaptive differentiation. So for this chapter, we're going to look only at one of the lynx species, which is the Eurasian lynx. I've drawn the distribution here because it's interesting for a few reasons. First of all, you can see it's a very wide distribution, and so it encompasses many different habitats, from the northern parts where we have tundra and taiga, through the boreal forest, also and arriving also to deserts, Mediterranean shrublands, coniferous forests. So there's a wide variety in the biomes in which these animals live in, but also if you look at the distribution as well, we can see that as I mentioned before, the westernmost part is very fragmented, and so many of these populations have been isolated for some time from the rest. So in a recent study from our group, they tried to reconstruct the relationship between different populations of Eurasian lynx, which I've drawn here with different circles for each sample. And what they found was that there are 3 main lineages of Eurasian lynxes. One is the one here in the east, which is in warm colors. There's another lineage in the west, which is in colder colors. And also there is a distinct lineage in the Caucasus. So this was the only kind of limitation the study has, is that it's based only on intergenic neutral regions, which means that selection is not acting on any of the the genomic regions that were used to reconstruct these relationships. So what is left to be asked is, what about what about selection? What about functional variation? Does any part that has a specific function is being selected upon, and if the selection will affect the structure that we see on the neutral part of the genome? So to answer these questions, we first had to identify the positively selected areas of the genome. Then we should characterize their functional annotation, which is a kind of fancy way to look what genes are involved in these selective processes. And also in the end, we wanted also to determine the effect that this local adaptation might have on the structure of populations. So to do that, we use a data set of 105 whole genome sequences across the range, and a total of 12 populations, which range between 6 and 13 individuals. For the selection scans, we only used 8 of the populations because of these isolated population in the West might have excessive amounts of genetic drift, which might confound the signal of selection we're looking for. So we decided to exclude them from part of the analysis. Then we aligned our sequences to the Canada Lynx reference genomes and extracted a final data set of around 2.1 million SNPs. So with this data set, we try to estimate selection. So assign a particular area of the genome as being selected for based on 2 different methods. So on one side, we decided that the areas that show the most amount of differentiation across populations might show this level of differentiation because of selection. So we looked into that, and also we decided to look into areas of the genome which most strongly associate with a particular genetic environmental variable. So we looked at different environmental variables and see which areas of the genome most associate with them. And this is called a genome environment association analysis. So what these 2 different methods give you is basically you have the list of all your SNPs you used, and each SNP as a score. This score is representative of how likely it is that this SNPs is being selected for. So you can try to average this score across close by SNPs to try to create genomic windows which have a similar signal and say that the most differentiated or associated windows are the ones that are being selected. So you have a final list of candidate windows for selection. So with these windows, you can look at 2 things. First of all is functional annotation, which, as I said, is looking at what genes are in these windows which are being selected. So to do that, as we used a lot of different variables, we wanted to try to average the signal of different variables to group the windows into window groups which show similar signal across environmental variables. That means that if we build a heat map with the columns as being the different genomic windows and the rows, the different variables, we can see how this group of windows, for example, is grouped together because they respond similarly to this group of 3 variables, or this different group of windows is responding especially strongly to this variable in particular. This gives us a way to group different genomic windows and based on their signal similarity. With these different genome groups, window groups, then we can look at what genes are present in those windows, as well as the ones from the highly differentiated part of the study, and make a list of genes. And with this list of genes, what we've tried to do in the future will be to see if there are any specific functions of the genes which are associated to the specific variable. So what is the effect that the genes have on the organism, and why is it being selected for? On the other hand, we can take these genomic windows which are being selected and look at the patterns that it creates in the population structure. How do we do that? We simply build a principal component analysis with the different individuals from the different populations and compare neutral areas of the genome with, for example, the over-differentiated regions. And what this kind of comparison gives us is a kind of a signal that the differentiation we see in the neutral genome doesn't appear to be present when we look at selected areas. So on one side, and also we are looking at there is a sort of start of formation of structure in the in the eastern clade, which is this one, which is neutrally very homogeneous. But in the selected areas, we can see that this pink population from the Vladivostok area is starting to separate. So this is based on the over-differentiated regions, but we can see this kind of pattern also looking at the areas of the genome associated to different environmental variables. So this is a very similar graph, only that we added the axis of the different environmental variables as well. So this is called a redundancy analysis. And on one side, we have the neutral genome areas. And on the right, we have the areas associated to different environmental variables. So what do we see here again is some kind of homogenization in the Western clade, which calls the attention to that maybe this kind of differentiation is only caused by genetic drift and not by a different selection. And also the formation of structure in the eastern lineage. Sorry, in the eastern lineage, we see the kind of starting a formation of structure with these two different populations starting to separate from the rest, based on their response to two different environmental variables. Bio5 for the Yakutia population, which is the temperature in the warmest month, and Bio1 for the Vladivostok population, which is the mean annual temperature. So these two populations appear to be responding differently from the rest of their group based on these variables. So general take-home message we have so far from what we found so far is that there is evidence that the differentiation observed in the Western lineage is caused more by genetic drift than it is by selection. And also that the divergent selection is starting to act into the eastern lineage to form different groups based on different environmental pressures. We still have to know what genes are involved in this kind of patterns. And so that is still up to know. So to give a final summary of what I'm gonna be doing in my thesis, in chapter 1, I will try to reconstruct the evolutionary history of the genus using models that include information regarding demography, differentiation, and migration. In chapter 2, I will focus on analyzing the patterns of local ancestry and introgression along the genome. And in chapter 3, I will try to define adaptive and functional differentiation among Eurasian lynx populations and try to analyze the effect that this has on population structure. With that being said, I would like to thank everybody who comes from my group and also all our collaborators from outside of our group, and also our center for providing very wonderful services, and all of you for listening to me. Thank you.
Thank you so much, Enrico. It was super cool having your talk. I mean, I'm impressed because it's a huge amount of information you are handling. So you didn't tell that you need to have like a programming skills for this. And I mean, I know that this is a hard work. So, and I also congratulate you because the presentation was very clear, very well structured. So very well done. So I don't know if people want to ask things in the chat while they are. So please ask your questions in the chat of YouTube. I have several questions, and also my colleagues, Agus and Mari, they are gonna join later and ask more. Okay. Because I don't work in genomics at all. Okay, I'm gonna ask you very basic questions, but I feel a little bit ashamed of doing that, but I hope this is gonna be helpful for the others that they don't really work on that. So I want to understand better, how do you really work on this environmental variation of the genomes? So you said in your third chapter, no, that you are looking for the genes that are involved in the environmental response.
Yes.
But you are not really. I mean, I don't understand how you know that these genes are really responding to that variables. I mean, I want to. My question is related to. So, which kind of response are you having? For instance, you were saying that some populations are separating according to the response to the mean temperature. So, yes. Are these populations they like more warmer temperatures, or I mean, I would like to understand better how you are doing that and what are the consequences, the implications for the ecology of the species, the populations.
Yes. I think there's you highlighted a big limitation that we have, which is basically we're looking at genomic patterns, no? So we look at the genome and we can look at how different areas of the genome are more correlated with different environmental variables. But we don't have much information regarding the phenotypes of the individuals we are using for this kind of analysis. So we can say that maybe these parts of the genome are responding to this particular environmental variable, and especially for this population. But until we have some kind of study which tries to link the genetics with the phenotype, like the genotype with the phenotype, we won't be able to really assess what is going on. But the only trace of information we can have is what I talk about here, which is functional enrichment analysis. So basically, if you say, okay, these group of windows appear to be responding to the number of snow days there are, okay? So you can say, okay, in these windows, we find these 80 different genes. Of these 80 different genes, most of them are related to body size. I don't know. I'm just like kind of freestyling, no?
Yeah.
But if the most of these genes are involved with body size, you can imagine that the adaptive response of these populations is to have a change in body size based on the number of days of snow during the year. So this is the kind of inference you can make. The big limitation is that you don't have anyone in the field looking at lynxes and says, okay, they are actually bigger, or they are actually smaller. But
so that is my second question. You don't have field data, no, from these populations.
We do have a collaborator which is working a lot on trying to build this phenotype kind of relationship with geography. But the other limitation is that this kind of population data doesn't translate into the individuals we actually use. So even though I have a table of the mean body size for the different populations, I don't have the mean body size, the body size of the individual I used for this study. So there might be some kind of error included in that, but it is something we are looking into. One of the collaborators we have is very interested in especially in the fur color patterns. And so how are these differentiated in different populations? But this is kind of a different field of study. So in that kind of study, you're trying to either relate the phenotype with the environment or the phenotype with the genotype. But in this study, what we're doing is trying to relate the genotype with the environment.
And it's only a statistical correlation at the end, no? So
yes, it's not. I think the model is based on it's a bit more complicated than a simple correlation, but yes, in the end, yes.
Okay, thank you. I think Agus has a question.
Yeah. On that line on relating the genomic regions to the genotypes and then to the phenotypes and the environment, I would like to know whether these regions that you use in your analysis, they are mapped to specific regions of the genome. Like you can tell where in the genome are these SNPs that you are using located?
Yes.
Okay, so then it's easier to find the genotypes, the molecules.
Because the way the pipeline works is that you have a reference genome, which is annotated. So you have we use the Canada Lynx as a reference genome. So you have a genome of a Canada Lynx, and it tells you, okay, from this base pair to this base pair, you have this gene. And from this base pair to this base pair, you have this other gene. And then you have your alignment. So your individual is all of this sequencing reads are mapped to this reference genome. And so any information you have you can locate it on the reference genome to say what is there.
Okay. And the map is related to a number of pair of bases. It doesn't have into account. Does it have into account the chromosome in which?
Yes. So in your reference genome, you have your coordinate, like a coordinate system that tells you, okay, from the base 0 on the chromosome. So it's always like chromosome position, and so you always have the information also on the chromosome.
All right.
So you could ask questions regarding if the signal is based on some chromosome in particular or not. It's especially interesting for looking at patterns of for example, the sexual chromosomes. So that might be important, no?
Okay. Maria also has a question.
Well, more than a question, is kind of like a comment regarding what you were saying, just to clarify. So you were talking about ecology when you were asking the question. This kind of analysis doesn't look at ecology. It looks at the genetic signature of selection that depends on environmental factors. So it is a different approach, like he said. So you are looking for signatures in your genome, and you are associating those signatures for certain environmental factors.
Yes, it would be the perfect triage would be to have the whole picture, but it's also very complicated.
Yeah, so the other part of the ecology is, like he said, you will have to have the phenotypes and stuff. I have more questions, but there are also questions in the chat, so I can wait. So
go ahead. And then we will do the question.
My question is regarding your ABC modeling from your first chapter.
I'll go there. You can start asking.
Okay, I have two questions actually. One I'm curious to know which were the modifications that were done specifically for your, because you said specific modifications. Yes. So that's a curiosity. And my second question regards the models that you were testing in particular, right? Because I don't know if I misunderstood something, but intuitively I would say that I would also test for the admixture that you were saying before. And I don't know if, like when you were saying that Lee 2019 was saying that there was admixture, right?
Yes.
This one. So you are not, are you testing this or not?
Yes. So I'll answer it backwards, okay? So all of these models, we're trying to be more, I don't know if it's conservative or crazy, but we put introgression events across all of the lineages. So admixture is possible for the model for all of the lineages. Because the model also gives you the parameter at the end, which is the migration rate or something like that, then you can say, okay, if the model is inferring a migration rate of around 0, then I can say, okay, yes, I tested for this, but it didn't happen. No, but we included migration events across all the lineages.
Okay, so instead of doing pure divergence, founder, whatever, admixture, you are putting the introgression events within your models, and then by looking at the parameters, you will see.
Yes, basically because the model has this advantage that even if you put an event there, then it estimates the amount of that event happening. Yes, and so if it's around 0, then it's just 0, and then you don't lose any. Like simplifies a lot the process, because if not, you would have one model with no introgression, one model with one event of introgression, another model, and it would be very tedious on on a computational
Yeah, I
side.
guess that adding the deep learning to
Yes.
the traditional ABC kind of helps
Yes.
and changes a little bit what are your expectations and how to build the models.
So basically, the implementation they made was usually you would jump from here, from this part where you simulate the data for the different windows.
Yes.
You would just get an average summary statistic for all the windows.
Okay.
It would jump this middle part, and so even there is only one deep learning algorithm going from this stage to that stage. Mm-hmm. What the introduction of building a distribution of all the summary statistic across all the windows gives you is the possibility of different windows having different signals.
Okay.
So I have actually, in the end, I knew something was coming about this, and so I have a slide about this.
Oh, that's nice.
It's here. So you have your different models, and this first step will give you a distribution, which will be different based on the different model you're testing. Exactly. And so from this distribution, then you get a summary statistic, which takes into account, I don't know if this has a long tail or a short tail, or this kind of information is summarized as well.
Okay, nice. Pretty good.
Enrico, so Agus has a question, but there is another question in the chat. Agus, can you wait for the question in the chat, or?
I could, but it's just because it is pretty related to what we're talking about here.
Okay, go ahead.
So very briefly, in order to differentiate between these models, I guess that you need a minimum sample size. But I am a bit confused about these sample sizes of genomic studies, because on one hand, you have like millions of SNPs, but then you have a very few numbers, very small numbers of populations.
No, individuals even.
Or individuals per population, even less. So
2 individuals per species.
How can we, is there any theory or any principle that we can follow to assess whether your sample size is good enough for what you're doing or is not?
I think the general idea is that okay, so basically the thing you're saying is that you don't know if the differences you see between two species might be the same amount of differences you see between two individuals of the same species, no? That would be a problem in sample size, no? If you're
exactly
saying,
that, like as you increase the number of individuals for each species, the amount of variation, of genomic variation could increase, and then the difference between the species could be blurred or otherwise. You know,
I think the
striking.
The general assumption is that the differences within a species are much smaller than the differences between species. So if any pattern you're detecting, if you're specifically looking for new species, this might be a problem. No, you need a sufficient amount of information to distinguish, oh, this is a different clade from this other one. But when you have established clades like these ones with different studies certifying their level of clades, using also outgroups to define within the clade, then you can start saying, okay, the differences I detect between these individuals of this species and the species 1 versus the species 2 is going to be much bigger than the difference I might see between one individual of species 1 and another individual of species 1. And so then you can start saying, okay, any individual of species 1 would be enough information to compare with species 2 because they are so different between each other. You know, like it's like if you have no, I don't know how to explain it, but if you have a tree, no?
You can explain it with the graphs, with the PCAs that you have, because
yes.
you have your individuals within the same species grouped together.
That's
perfect. All that you are looking at.
So
more or less.
Even though, yes. So the differences you might find in this blue population.
The blue populations, each point is an individual or a population?
It's an individual.
Okay.
So this blue population here is relatively close to each other, no? They are relatively homogeneous. So any difference I want to see of blue versus pink, it's gonna be more or less the same. It doesn't matter if I pick this blue point versus this pink point, or this other blue point versus this other pink point, because the main thing I'm looking at is the amount of difference there is.
Mm-hmm.
If I'm comparing across these two, then I might have a problem, no? Because this one is much more distant to this one. I don't know if I'm pointing at,
yeah, or for example, the light purple ones with all the red, oranges, and pinks would be pretty similar in axis PC2, right?
In PC2, yes. But this is, PCA is a bit misleading in this sense, because it tries to find a perpendicular axis of variation. So once you've detected this, you're not gonna detect it again in PC2. So it's a bit misleading in this sense. If you look at it as a tree, and the only axis important in the tree is the PC1, then you would say any individual of this blue group is equally distant from any individual of the pink group. So if I pick one and look at the differences with any one of the pink ones, I will have a big enough picture of the story. That's not to say that having more samples is better. I think that one of the things I tried to mention, I don't know if I was clear, is that we're gonna use 8 samples to try to test the model and see how it works. But we're gonna run repetitions with all the samples we have to try to see if it's consistent enough.
All right.
Yeah, there is someone waiting in the chat, so I will ask. So it's Tere. Sorry, Tere, for being so slow asking your question. So Tere, she says, super cool, Tere Boquete, super cool PhD project, Enrico. I loved your presentation. Thanks. I'm curious about the ghost population lineage in your demographic models. Do you have any evidence that this lineage existed, or because you don't have, and she said, second, because you don't have genomic data, how do you include in the models for the, sorry, it's the second chapter, no?
Yes, I went too far. Thank you, Tere, for the question. So basically, the ghost population was a result of a few things. So we have the new implementation of the model, which is something quite new for us. So it came like a month ago or 2 months ago. But this part of the project was started around 1.5 years ago. So in the early stages, we had some preliminary results, which indicated that any model with a ghost included would have higher support. Okay, so the question remains, why did you include a ghost in the first place? So the ghost was included in the first place because we were trying to explain a few different things. So first of all, you can imagine that with how the fossil record history of the lynxes in Europe in particular it appears to have been that the Iberian lynx was much more spread out in Europe and not only limited to the Iberian Peninsula. So it might have been in contact with any lineage of Eurasian lynx that was there. But also, if you look at the fossil record, you can see it like a double event of coming into Europe of the Eurasian lynx. So you can imagine if there is an ancient lineage of Eurasian lynx or any of these ancestors of these lineages living in Europe, it might have come into contact with the Iberian lynx in some moment. And so what this might also help to explain is why the signal in the low recombination areas of the genome it gives you this story, which might be the actual the story before the high amounts of introgression. But if these high amounts of introgression have come not only from the Eurasian lynx, but also from an ancestor of these two, or an ancestor of the Eurasian lynx inhabiting the Europe during those times, it might also be an additional confounding factor. So the idea of the ghost population is a mix between the geographical location of the Iberian lynx in ancient times, which was much more expanded in Europe, and also the fact that Eurasian lynxes appear to be coming into Europe in different moments, and also the fact that the actual genome of the Iberian lynx might be the result of the mixture of these two lineages, which would put it even more closely to the Eurasian lynx.
Okay, but you didn't have any fossil evidence of this ghost population?
No. It's. I think that the inclusion of ghost populations in general is something kind of new, but it's also something that kind of makes sense. So it makes sense that during evolutionary history, you have a lot of lineages that have disappeared. So you can imagine like extinction taking place. And so it makes sense that any lineage that you can try to trace back with these kind of models would leave a small trace in your genome. And so that there is a boom in this kind of ghost population inclusion in these kind of models. So I think it makes sense on an evolutionary point of view. And I don't know. Let's see, maybe it doesn't have any support, then it will be discarded.
But yeah, they make sense. Also, they've been using just in population structure. When you don't know if you have sampled everything, and sometimes population structure analysis give you weird results that are only explained by a ghost population. Mm-hmm. So it has been used.
Because of course, and yes, so you could have a sampling. No, you're not sampling everything that you are, even if though it's not extinct. No,
yeah. Mm-hmm. But it's really cool that you can actually include those and test that in your models. I think this chapter 1, it's really nice. The combination of the traditional ABC with deep learning, it's going to.
Yes, I think we're very lucky to be collaborating with these kind of people, because it really they're like magicians of the.
I have a question before, if anyone else has another question in the chat, or Amadi or Agus, but I would like to, because for me it was not clear at the end if either the Iberian lynx is more related with the Eurasian or the Canadian. So what do you think? What do you finally think?
You and me both. I don't know.
Don't know, no?
No, I think this signal that they found in 2019 is pretty solid. So I in my mind, and also correlates very good with this one, which is mostly based on fossil records. So in my mind, like this is in my head, this is the truth. And so what we try to do in our models is try to be as closely reproducing this as we can, and see if we actually get enough support for it. The other thing is that the actual average signal across the genome is this. So there's kind of a you can't debate too much with data. So if the data is telling you something, it might come from any different process, but the data is telling you something. So it might be possible that our model isn't able to reconstruct this event with the low recombination areas of the genome or anything like that. Our hope is that if we include enough admixture in our model, then this model would be more supported. But it's not a given.
So, and how do you explain according to the distribution? Can you put the map of there was a beautiful map at the beginning?
Yes.
The map of the distribution of each species.
This one, no?
Yeah, or the other one. I prefer the other one.
You like it? Okay.
This one. Because it's really, I mean, you can see how close they are, no? I mean, I can understand that the Canadian and the Eurasian lynx are related because they are, I mean, there is a varying, maybe during the glaciation or so on, where yes. A communication for the varying is strict. But for the Iberian lynx, I will think that maybe the easiest explanation is they came from the Eurasian and they rested isolated of how, but they are not. In fact, if you are supporting the second theory that the Iberian lynx is not related with the Eurasian, they came from another way. No?
But it's not that it's not related. It's just the moment of the relation ends before the one with the Canada lynx. So if you imagine, it's not that they're not related. So the picture is the bobcat goes into America first, no? If they're coming, we don't even know if they're coming from Eurasia or from America. So I don't know if the bobcat went to America first, or there were lynxes in America that went to Europe in one big event. So the bobcat separates first. Then you would have this scenario in which either you have this separation between Eurasian and American lineages, which would be this is too far, but it would be with these two separating first and these two separating last, no, like you're saying. Or you could have an event where this separates, then the Iberian lynx is isolated in Europe and separates from a lineage which was not present in North America, but only in Eurasia or in Asia, of the ancestor of the Eurasian lynx and the Canada lynx. And after the Iberian lynx is separated in Europe, there is an event of migration through the Bering Strait into America again, separating the Canada lynx from the Eurasian lynx.
I see. So, but that means that the Eurasian and the Iberian lynx has not been mixing since super long time, no?
The Iberian and the Eurasian lynxes.
Yeah, if you support this hypothesis, this last hypothesis,
the idea is that there might be a moment of separation which causes the divergence, but this moment of separation might not have lasted long through time. It might have been, I don't know, I'm not gonna say number because I'm gonna say something that's not true. But if you imagine there is a separation which causes differentiation, but then this barrier is broken somehow, then you can still have gene flow between these two lineages, even before that the Canada lynx lineage is formed.
I see.
And this is actually what the fossil record is kind of reconstructing the story I'm telling you. So what the guy who reconstructed the history of lynxes from fossil records said, they came from Africa and then they entered Europe and Eurasia. And then and then there was like a panmictic species across the whole northern hemisphere. But then the separation of the bobcat came like 3 million years ago, around 3 million years ago. And shortly after, Western, so like Europe and Asia separated, and in Europe there was the Iberian lynx. And in Asia, there was the ancestor of the Canada and the Eurasian lynx. Then around like 200,000 years ago, there was a migration event into Canada from the common ancestor of these two. And so this is a much more recent lineage. This is what the fossil record is kind of telling. Still, you have the bias in the fossil record that you might be missing one fossil, which is changing all the history you're finding.
Wow, but it's amazing. It's very good, super good. Thank you so much for the explanation. So I don't think there are more questions in the chat. I really enjoy your talk. Thank you. Enrico was super, for me, super explicative. So very, very funny. I don't know if Maddie or August has another question.
No, I don't.
Neither, I don't have any more questions.
So thank you so much, Enrico, and congratulations again for your work. And so next week we will have Jesus Manuel Diaz Fernandez for he's another PhD student, and he will give another seminar about birds this time. So thank you so much for being all you here, and see you next week.